Welcome to your lesson on DNA structure and chemical composition. Hopefully some of the stuff is not new because we did cover it in the first unit, but there's a lot of stuff that is new. So let's dive in. Now, just a reminder, some of these macromolecules are polymers. Polymers have monomers. Polymer just means there's many of these little monomers stuck together. The subunit or monomer of a DNA chain or a DNA polymer is called a nucleotide. T for 3. There are 3 parts. You've got the 5-carbon sugar, which is this guy right here. 5 carbons, not 6 like a glucose, but 5. And just a reminder that DNA has a deoxyribose, so it's missing an oxygen, compared to RNA that has the normal ribose sugar. And this is the spot right here, right at the 2nd carbon. That is the difference between the two of them. I don't need you to know where the difference is. I just need you to know the deoxy versus the regular ribose, and then you should be fine. When you see 1 with the little tick mark, it's read as 1', 2', 3', 4', 5', and so on. Okay? So just remember that. So again, 3 nucleotide, T for 3. There is a 5-carbon sugar, depending on whether it's DNA or RNA. There's a phosphate group, and you can see that attached right here. It's always attached to the 5' carbon, and that's going to become very important when we start talking about DNA replication and how you actually stick the strand together. So always at the 5' carbon, and then always at the 3' carbon, there's a hydroxyl group. This is very important because we're going to be connecting the next nucleotide right here. So the phosphate of the next nucleotide clicks in at this spot. And then on that nucleotide, another one clicks in the bottom there. It's going to be very important. 5' versus 3'. This is where we're always adding the new one because of the open hydroxyl group. Can't say that enough times. The third part would be the nitrogenous bases. You've got thymine and cytosine, so T and C, and you've got adenine and guanine. You've also got a uracil in place of thymine for RNA. Notice there's only 1 carbon and nitrogen ring here. 1 carbon nitrogen ring, and then there's 2 rings over here. So you've got pyrimidines are the single rings, and then the purines are the double rings. This is the silly way that I remember it. To me, when I see the word purine, I think Purina cat chow. And when I think cat chow or dog chow, I think, hey, they need to have a water bowl and a food bowl. So to me, it makes sense that purines are a double ring because you got to feed the cat. Whereas over here, pyrimidines are only 1 ring. So that'll help with the double ring versus single ring. The way that folks try to remember which ones belong to purines and which ones belong to pyrimidines is look at the Y here. Y in pyrimidine, and there's a Y in thymine and a Y in cytosine. And then for me, when I see double ring and I see a G, maybe it doesn't look like it to you, but can you see there's a ring there on the top of the G, and then if you imagine it, a ring where it hooks down? And the same thing if the A is written like this, you get a top ring and a bottom ring. So those are my silly ways of remembering what's what. So Purina cat chow with a double ring on the G, and then pyrimidines have the Y's, and that helps. So trying to commit those to memory so you know which is which. I also mentioned this, but now we're getting into more detail. You always have T bonding with an A, and you always have C bonding with a G. The only time that A doesn't bond with T is if you're dealing with RNA, and RNA, instead of having T's, has U's. So you'd expect A to be attaching to a U here instead of a T. The only difference between a thymine and a uracil is this little group right here. Instead of having the CH3, uracil has an H. So it still makes a great bonding partner with an adenine. Okay? So T always bonds with A. That's very important you know that. When they bond, the way that they line up, they have 2 little hydrogen bonds. Now, I want to remind you with this little graphic over here, the bases themselves, that's what these little rungs of the ladder represent, they don't actually physically touch one another. Instead, they have an attraction between each other, and those are the hydrogen bonds. So this is where you've got the nitrogen at some point in time is stealing the electron for part of the time. Even though it's covalent, it's still polar covalent. You've got this hydrogen's electron being stolen, so it's slightly positive for a time. And then this nitrogen, who is slightly negative because it's just its nature, it's going to want to pull on that positive. So you got a negative charge across from a positive charge, and they have this pull. So it's not a real bond. That's why it's sort of squiggly here. It's not very well drawn. And then over here as well, this oxygen is definitely stealing electrons from this carbon that's hiding here. So it's slightly negative. This guy is slightly positive because of nitrogen's pull of its electron. So again, you've got a bond here and a bond here. The key thing to remember is that there are 2 hydrogen bonds between the A and the T. That's why I've written it with the 2 lines here. And then 3 bonds between C and G. 1, 2, 3. That's why I've written it 3 like that. Please memorize that fact. It's going to help you so much later on if you remember that. The way that it sort of works out for us then is if you look at this, you've got 1 and then 2. So as far as how wide this would be, it should be 3 rings wide. Same thing with this guy. You've got a single pyrimidine with the double-ringed purine, so it should be 3 wide. If the helix happened to have 2 purines paired together, and this is the type of question that ends up on the test, 2 purines together, well, the purines are the double rings, you would actually have 1, 2, 3, 4 rings wide. Okay? If that's the case, then you'd see a bulge or a convex section. This would bow out. And if it was 2 pyrimidines, only 2 single rings, well, it should be 3 wide, you're going to see a dip or a concave section. Just a reminder as well, as far as the language goes, T for 3, base, the 5-carbon sugar, and the phosphate. If we're talking nucleoside, I think S for second or 2, and that's just the base and the sugar, no phosphate. And of course, we have to talk about the names of bonds. So the name of this sugar here would be deoxyribose because it's missing the oxygen here. So this is going to be a DNA nucleotide. And at the 5' carbon, you've got the phosphate group. At the 3' carbon, you've got the open group ready to receive the next phosphate coming in from the next nucleotide. And then at the 1' carbon, you have the base attached. So the bond that's formed here between the 5' and the phosphate is called a phosphodiester bond. Memorize that. The bond between the carbon and the nitrogen, or the nitrogenous base, the entire thing, this bond here is called glycosyl bond. Don't confuse this word glycosyl with all the other glyco and gluco words you've had to learn so far. Make sure you remember glycosyl is this bond here at the 1 carbon. When we click in and get the next phosphate group clicked into the OH, we'll have another phosphodiester bond, but I'll show you that on a picture later on. We jump into Chargaff's rule. Super simple. If the proportion of A equals T and the proportion of C equals G, because these guys are paired and these guys are paired, then the number of purines that you have should always equal the number of pyrimidines because they're always paired with each other. You have a purine and a pyrimidine, pyrimidine, purine. Okay, so these numbers should be equal. That means that all the A's and the G's should be equal to all the T's and the C's, which means that if I know that in a chemical strand there's 40% adenine, by the way, another test question, there's 40% adenine, what percentage are the other 3 bases? Well, if it's 40% adenine, then that means it's 40% thymine. That means that 80% of this DNA strand is already made up of these 2. There's only 20% left. Therefore, 10% must be cytosine and 10% must be guanine. Done. So there's another little easy type of test question right there. Okay, let's get into some of the history now. Now, how do we know it's a double helix? Where did all this kind of idea come from? Well, if you remember back in 1952, you had Hershey and Chase who finally and elegantly proved that DNA was Freddie Griffith's transforming principle. Well, a year later, you've got Rosalind Franklin, who has been for quite a while working on this version of X-ray called X-ray diffraction. You do a lot of this with proteins to figure out what the structure of protein was, to figure out whether it was a helix coil or a beta-pleated sheet and stuff like that. And she did it with DNA, and she was working really hard on this kind of stuff. But she didn't know that a colleague of hers in her office had gone to a conference, and he had taken her picture with him to the conference, and it just happened to be that James Watson and Francis Crick were there. So here they were working on DNA, trying to figure out what the structure of it was. And when they saw Rosalind Franklin's picture, again, without her knowledge, I know it doesn't look like much to you and I, but if this is the way that the X-ray diffraction turns out, it's evidence of a helix coil. So then they immediately jumped into work, and by 1953, they had done some calculations, and they had figured out the diameter, the number of base pairs, what was logical for the distance between the pairs, all that kind of stuff, all, again, based on this right here. But I just want to mention, 2 nanometers wide, 3.4 nanometers per turn. Now, remember, nanometer is a billionth of a meter. But then when we get down to the distance between the bases, that's even smaller. It's 0.34 nanometers. So people stop liking to use billionths, and they want to use 10 billionths. So an angstrom, it's this A with a little circle on top. The angstrom unit has 10 of these guys per nanometer. That makes it a 10 billionth. That's super small. So you can either write the distance between bases in nanometers, or you can write it in angstroms, either way. This would have made more sense if I was actually teaching you in person, but DNA usually turns clockwise, which is right-handed. So if you could, stick your hand out right now. I know it seems silly to do that to a video, but stick your hand out right now, face a clock on the wall. Obviously not a digital clock, friends. Make sure it's a real, actual analog clock. And then take your thumb, put it upright so that it's the top of your hand, and then turn out towards your body. That's called a right-hand turn. If you can imagine now that you are spinning around the outside of DNA, okay? And if you notice your hand is moving clockwise as your right thumb moves to the outside of your body. If you do the same thing with your left hand, stick your hand out, make sure your thumb is on top, and then you can turn your thumb to the outside of your body. That's called a left-hand turn or a counterclockwise turn, because you would be turning counterclockwise to what a clock would be. And some DNA spins in that direction, but most DNA is right-handed. Most spins in the right direction. That's not so important for our course, but it will help you if you're doing anything with DNA coming up in your university classes next year, which is very exciting. You guys are almost there. Let's jump down to this diagram on page 4 so I can show you what all these little numbers look like. Okay, so here we are. So here's our helix coil. Helix meaning how it's spinning, okay? And this one is right-handed. You can see it's spinning towards the right. Their calculations let us know that the width of it was 2 nanometers, and that from this, notice how it repeats here. If you follow the purple strand, you can see it bows back out here, kind of like a nice little sine wave. So it bows back out at this point. So this is one full turn between this location here. You'd have another turn here between the blue and the blue over here, just like normal waves, like looking at the crests, 2 crests of a wave. This is their wavelength if you want to think of it in those terms. 10 bases per turn. And that entire distance is 3.4 nanometers, which means that the little distance in between these bases must be 1/10 of that, or 0.34 nanometers, or 3.4 angstroms if you don't want to use the little decimal numbers. Okay? Memorize these dimensions. It's going to matter. So 2 nanometers, 10 bases per turn, 3.4 nanometers per turn, 0.34 or 3.4 angstroms per the distance between those two. Okay, and I'll accept either of these answers as we're going forward. So again, you've got the 5' carbon. This is where you're going to bond the phosphate. You can see that here. You've got the 1' carbon, and that's right here where you're going to bond the base, and that's on every single one of these sugars. And then you've got the 3' carbon with the open hydroxyl group. It's not opened here anymore because we've clicked in the next phosphate, and then here again is the 3' carbon, but you've clicked in the next phosphate, and so on. So it's only at the end of the line when you see the open hydroxyl group ready to be available to pop in another phosphate. You cannot pop in a phosphate at the 5' end down here or the 5' end up here. There's just no space to attach a phosphate to a phosphate. So it's always the open hydroxyl group, the way that the enzyme works that joins them together, always pops it in at the 3' end. That's why we're going to say that the DNA runs from 5' to 3'. And you're going to see that when we do some replication, because you're always adding in this direction. Okay? Now the DNA code, as it says here, is read from 5' to 3' direction. We call that the sense strand. You're going to see these terms over and over again, and by the end of our next lesson, I'm hoping that sense, coding, and non-template all make sense to you. For now, I'm just going to say it a million times so that you guys can hear it. Okay, so the one strand will be called the sense strand, and then because the other strand is exactly opposite to it, it's called antisense. So if you look at this here, you've got T, and then you've got A. Well, those are opposites. So if this is the sense strand, this has to be antisense, okay, because of the way that it runs. Antiparallel copy also runs antiparallel because you can see that if this is the 5' end and the 3' end, now this is the 5' end and this is the 3' end. So you've got it running opposite direction. So if the antiparallel copy also runs 5 to 3 but in the opposite direction, you call it antisense. You also call it complementary. So the antisense, because it matches, notice this is an E on complementary, not an I. please spell it correctly. It's not saying nice things to the other strand. It's matching the other strand. Complementary, not complimentary, okay? So it's not just saying nice things. So this guy, the complementary strand, is always going to be opposite to what's going on over here, okay? Two other terms we use would be coding, but non-template, and then non-coding, but is the template. And that makes more sense when we start trying to use the DNA to actually make RNA so that we can get a protein out of it. So these terms will come up more so when we're talking about protein synthesis in a couple days from now. All right, but for now, please know that one is sense and one is antisense. Yes, they both run 5' to 3', but only one of them is actually used as the code to make a protein. Okay, so here's an example again. You have sense strand 5' to 3', and notice it's exactly opposite. So if it starts off with ATCG on this one, it does not at the 5' end have ATCG. It's a completely different set of letters because you're always reading 5' to 3' direction. So if you try to read this one, it's clearly being written in garbled backwards. Because if ATCG means something, CAAT doesn't necessarily mean anything. So this is the one that makes sense. It's actually written to make sense, and this one is just the complementary strand running the other direction, okay? This one is coding because it matches the mRNA sequence that we're going to make in a couple days. The non-coding doesn't match it. Non-coding would be complementary to both the sense DNA, which is this guy, and the mRNA, but you'll see that, I promise you, in a few days it'll make more sense. Just remember from this lesson that the 5' always starts with a phosphate, and the 3' always ends with an OH, or an open hydroxyl group, okay? Again, the hydroxyl group has to be exposed and available to add the next nucleotide. That means that the strand's always going to grow in the 5' to 3' direction. This is another type of piece of information that ends up being test questions. I want to show you again the names of the bonds. You've got between the 1' carbon of this is one nucleotide up here and a second nucleotide here. The 1' carbon of this nucleotide is attached to the phosphate group at the 5' end of the next guy. So this here in red is a phosphodiester bond because it's between a phosphate and that carbon there. This one is also a phosphodiester bond because it's between the phosphate and the carbon here. So you have two phosphodiester bonds, this one here being within the nucleotide, and then one between nucleotides. Then you've also got a glycosyl bond, and that's between the sugar and the base. So glycosyl, glycosyl, glycosyl. Okay? And here's a nicer version of being able to see how it runs anti-parallel. So you definitely have the 3' group down here and the 5' up here. So you got 5' to 3' running in this direction, and then 5' to 3' running in that direction. You can even see the sugars are flipped upside down. Okay. Please go ahead and take a look at page 214 and write out definitions for genome and gene. I'm not going to add them to this note. Make sure you know the difference between genome and gene. Okay, final. We're just going to compare quickly the DNA of the prokaryotes. Reminder that prokaryotes are all bacteria. Whether they're the fancy archaeobacteria or whether they're the fancy eubacteria, doesn't matter. All prokaryotes are bacteria. All bacteria are prokaryotes. Okay. Now, they don't actually have a nucleus. That's what sets them apart. It's even built into the name. Prokaryo means there's no nucleus. But of course they have DNA. They're living organisms. They need to have a blueprint or a code to make more living organisms and to repair themselves and to grow. So instead of having an actual nucleus, they have a nucleoid region. It's basically just an area. There's nothing fancy to it, but it's an area where you're going to see the bacterial chromosome. So they call it a nucleoid region, and I've just dashed it out here just so you kind of get a clearer picture of it. Okay, so bacterial chromosome, they have one. You have 46 chromosomes, and yours are linear. They have one, and it's circular. Okay. Now, they only have one copy of each of the gene in that single circular chromosome, so we consider all bacteria to be haploid. You are diploid because you have a copy of chromosome 1 from your mother and a copy of chromosome 1 from your father. That makes you diploid, 2 copies of every gene. The copies could be slightly different codes from one another, which is why some of us have brown eyes and some of us have blue eyes and so on. But you have 2 copies of everything. You are diploid. These guys only have 1 copy of every gene. This is why they tend to mutate a little bit faster than what a human would do. Because if you damage that one little copy, then there's no backup copy. So if these guys get some kind of a DNA change, they're going to have a DNA change in the protein that they create as well. So
Now, what's written on these chromosomes? What's the point of this chromosome? Well, this is all their essential genes. These are the genes that say, be a bacteria. Here's the protein that you need to create to be a bacteria. Here's how you grow. Here's how you reproduce. All those kinds of things, those very essential genes. Okay. Along with those genes, they also have sequences called operons, which we'll talk about in a separate lesson. And basically, the operons are on-off switches that say whether the gene is supposed to be used or not. We would say that the gene is expressed if we're going to make a protein out of it. So these are on-off switches. These help them to not just be making everything all the time, because then they would be wasting energy on products they didn't need, but to tell them when to make a product and when to stop making a product. Even in DNA, there's lots of regulation. It's actually really, really interesting stuff. Okay, in addition to their one circular chromosome in the nucleoid region, they also have these little plasmids. Now, plasmids are extra little pieces of information. They're non-essential, so it's not what makes a bacterium a bacterium. They can do without them, but sometimes they code for things like antibiotic resistance. So there might be a special section of it, a gene on there, and it actually gives them a little bit of an advantage because maybe it produces a protein that allows them to resist being killed by the antibiotic that you just put on them. It might give them just that little bit of an edge. They can last just a little bit longer, and if they can outlast the antibiotic, then they will have benefited. So this is what it actually looks like in an electron micrograph picture. This is a little circular small ring of DNA. What's tricky about the plasmids is that once they've got one, and they've got a little bit of a added bonus of information, these little tricky bacteria, they can pass it between each other by something called conjugation. They do that by having the two bacteria line up. They form this little bridge between their cell membranes, and then the little plasmid gets copied by the first guy, and it just gets transferred to this guy. So now they both have the same information. And again, that's called conjugation, and you conjugate via a pilus bridge. This is very annoying when you're trying to fight a bacteria. This is what helps to make superbacteria because they share the wealth with one another. But it is extremely useful to us in the lab. The fact that they are happily going to share genetic information, that means that we can take this plasmid, we can alter the sequence, leave it as a little treat sitting there outside of the bacteria, and the bacteria will take it up and start creating whatever it is that we just put together. So this is actually pretty awesome in terms of biotechnology, that they like to share plasmids and that they like to use them. So we can trick a bacteria into making things for us. For example, we can chop this little plasmid open, and we'll talk about how we do that with biotechnology in a few days. But we can chop it open. We can take something like a human gene, the human gene for insulin, let's say, and we can insert it into this section here, cause the bacteria to suck it up so it becomes part of their regular working DNA, and all of a sudden, this bacteria is now producing human insulin for us. That's a game changer for people with diabetes, that now they can actually have a massive supply of insulin. You don't have to try to extract insulin from a donor. You can just produce it in the lab, which is what they do. It's pretty cool. So that's one of the great tricks that we have with using plasmids. Super useful to us. Now, they don't actually leave it in this messy lump that you see here in this diagram. I'm not sure why they ever created the diagram to look like this, but this is actually how they store their DNA. So when they're not actively replicating or actually making a protein, the chromosome is actually first looped, and these little yellow things are little proteins that are holding it together gently. They're looped like this little pretty flower, and then they're supercoiled. They're looped again. And then proteins are going to help bind and keep it compact so that it's not liable to just float away and be damaged by whatever, okay? So you want to make sure that it's nicely tucked away. And then when you need to create RNA from a certain gene, then you're going to just uncoil that one little section, use that DNA, and then coil it back up again to protect it. We are way more complex than that. Okay? The DNA of a eukaryote, which is you, a fungus, plants, animals, protista, those four kingdoms, the eukaryotes do have nucleus. They also have a heck of a lot more structure to their DNA. Now, the DNA molecule itself, as we talked about with Francis Crick and James Watson's stuff, was 2 nanometers wide, 2 billionths of a meter wide.
But that's not how your DNA sits inside your cell. This is extremely fragile and you want to protect it. So the first thing that you do is you take the DNA and you wrap it around 8 little proteins called histones. Together, the DNA plus a histone is called a nucleosome. And that's your first way to try and keep it safe. By the time you've wrapped it around the histone, you're now 5.5 times larger than you were here. So it's 11 nanometers now. Then you take the nucleosomes and you begin to coil them up. Well, now you've got what we've been calling chromatin, and it's 30 nanometers wide, so 15 times bigger than just your DNA molecule. Okay. Even now, though, at 30 nanometers, you cannot see this under a microscope. You could see it under a nanoscope, but not a microscope. Okay. So it's too small to be seen. So then this strand, you can see that this is just a portion of here, gets looped again. Okay. And now it's 10 times bigger than it was. It's 300 nanometers. Okay. Now, this only really needs to begin to happen. You coil it, and then you coil it again. Crazy, crazy, crazy number of coils. That really only happens when you're doing replication. So you would have already doubled up the DNA when it was loose. Then you begin to fold them. And by the time you're done folding them, you're going to have your 2 sister chromatids that are still attached at the centromere. Okay? Then these 2 huge, huge, massive, look at this, 1400 nanometers. Okay, 700 times larger and wider than just the DNA strand itself. Massive, massive compaction. So then you're going to pull this sausage apart from that sausage and away you go. The genes themselves in a eukaryotic chromosome, it's not evenly distributed. So you don't have, 30 genes per chunk of chromosome. It depends on the chromosome. Some chromosomes have more information than others. Some chromosomes are more important than others because of the information that they carry. Now, originally, they thought that the more complex you were, the bigger your genome had to be. So they thought that with humans being so smart and the pinnacle of all creation, how wonderful we are, that we must have the best, the biggest, baddest genome out there, until they started investigating plants. And they found out that some plants have a ridiculous number of chromosomes, way more genetic information than we will ever have. And then they thought, well, we would like to think ourselves as being smarter than a plant, so perhaps genome size is not linked to the complexity of an organism. Right? And you'll see that in your textbook. There's a few examples of organisms that are dumber than us for sure, and way more complex genetically than we are. So they had to let that go. And then again, the size as well. With the number of protein coding genes, this is the other thing. We thought we've got 100,000 proteins, so we must have 100,000 genes. And that actually turned out to not be true. When they did the Human Genome Project 20 years ago, they realized that, okay, we've actually only got somewhere between 25 and 30,000 genes. So somehow these genes that we've got must end up making more than one protein product. And I'll show you when we start our protein synthesis lessons how we think that might be accomplished. Okay. So in addition to that, not even every gene that we've got actually makes a protein product. So there's a lot of information we've learned since 1953. We realized that it's not as simple as we thought it was. It's not one gene per protein. There's something else going on. And we're still learning a lot about that. We're not at a final conclusion yet. We're still trying to figure out how all that works. Lots of great theories, and we think we might be right, but we thought we're right a lot of times that we weren't. Okay, so not all genes create a protein product. So that's a huge mystery. Why would those genes be there if they're not even making proteins? All right. There's also huge, huge patches of DNA in between genes that do code for proteins, and we're not 100% sure what those things are for because we don't see a protein product. So again, we got tons and tons and tons of information. We used to call those junk DNA, even back when I was in university. Used to call that junk DNA. And I remember back then saying, it's not junk, there's a purpose to it, we just don't know what it is yet. And sure enough, within a few years, people started calling it non-coding instead of junk. So lots of information. If this is a field that you're interested in, and you're interested in research, my goodness, there's still so much work to be done on DNA. This is the field to get into if you want to make your mark on the scientific world. All right, so let me encourage you with that. And that, my friends, is the end of this lesson.